Stephanie, can you hear me?
Yes, I can.
Good, 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 good. What was that? Okay, that was weird. You cannot minimize. Yeah, I get that. I don't want to minimize it. I want to maximize it. Okay, so. Stephanie, the road's pretty bad out where you live.
Yeah, like the apartments, they haven't even plowed it or anything.
Maddie lives in Jackson. She said the roads are rather impassable with the exception of I-94.
Yeah, I think 94 is okay now, I think.
Okay. I live off Teft Road and our road is not been plowed. It's pretty bad.
Yeah. Staying in today.
Okay. See Mrs. Schaefer take the quiz Monday. Try to get it
Okay.
done before Wednesday.
Okay. I can go in the morning before class.
Okay.
All right.
Yeah.
I took a lot of like.
Yeah. Okay. Yeah, just throw them up there, Maddie. Thank you. Very good. So if we take a look at our handout, again, this handout is on the Blackboard site, so you can download it yourself if you want. We talked last time about elongation factors. So here's how these elongation factors work. So let me emphasize, the ribosome has begun translation. You're past initiation. You have a methionine tRNA at the acceptor site, at the deletion site. You have an open A site. The two ribosomal subunits are together. The AUG is start codon is at the P site. And now you have an open A site. So the next charged tRNA is brought to the ribosome in bacteria by this elongation factor called EFTU. EFTU. EFTU is a GTP binding protein. So this complex of EFTU plus GTP plus the charged tRNA is brought to the ribosome. It will bind to the ribosome if the anticodon of this tRNA that is carrying the proper amino acid hybridizes properly with the codon presented at the A site. If it is, then this complex will properly bind to the ribosome. If it doesn't, then it'll kind of bind and bounce off. So only the complex of EFTU and GTP and the charged tRNA that properly corresponds to the codon that is presented at the A site will actually bind. When it binds, EFTU will hydrolyze its GTP to GDP and inorganic phosphate. Now the ribosome, the charged tRNA stays stuck to the ribosome. EFTU plus GDP floats away. Okay, let me say that again, or can you see that in the handout, or want me to go over that again? If the anticodon loop of the tRNA properly interacts, properly forms a nice interaction with the codon that is at the A site of the ribosome, if and only if those two sets of nucleotides properly interact, then the GTP is hydrolyzed, the tRNA with its charged amino acid stays, and the EFTU hydrolyzes this GTP and leaves. All right, good. The GTP is hydrolyzed into GDP and inorganic phosphate. Because bringing that charged tRNA to the ribosome requires the input of energy. Also, for it to be properly bound there requires the input of energy. The energy is in the form of that GTP, which is hydrolyzed. And remember, GTP is not hydrolyzed if that's not the right tRNA. If it's not the right tRNA, then it just bounces off and goes away until it is the right tRNA. Now, you have a EFTU bound to a GDP. It's not very useful at that point. Instead, there is an exchange factor called EFTS. EFTS is going to form a complex with EFTU bound to GDP, and it's going to kind of tickle it. It tickles the protein and it changes conformation, it spits out the GDP, and it takes up a GTP. So this type of protein is called a guanine, phosphorylated guanosine exchange factor, or a GEF, a GDP exchange factor. So it causes it to spit out the GDP, take up the GTP. Now EFTU is ready to bind to another charged tRNA. The GDP bound version of EFTU cannot bind to charged tRNAs. The GTP bound version of it can. Only by undergoing this exchange of a GDP for a GTP can the EFTU be able to bind another charged tRNA. And that exchange is catalyzed by this EFTS. EFTS.
So the EFTS will keep the GDP and then the EFTU keep
It binds to the EFTU and causes it to change conformation. That conformation has a very low affinity for GDP. It's kind of like when you got your little sister's toy, so she tickled you, so you let it go. You change conformation when she tickled you, right? Same thing with you. TS. In fact, TS, think. Okay. So it tickles the TU, and the TU changes conformation, and it lets go of the GDP, and the GTP now binds to it, and this protein then locks onto the GTP, and in this conformation around which it's locked, it has a very high affinity for charged tRNAs. It has a very low affinity for uncharged tRNAs.
So how does the EFTU get the GTP? Because if basically you're saying like the EFTS keeps the GDP, like the GDP.
No, doesn't keep it. It causes the TU to let go of the GDP. Doesn't keep it.
So then
It drops it, causes it to drop the GDP.
So where does the GDP go?
The GDP goes into the cytoplasm and it gets recharged by a variety of different enzymes. There's an enzyme called AWD that will take an ATP and it will phosphorylate the GDP and make GDP. So that you regenerate it, essentially. GTP levels are relatively high in the cytosol. They're relatively low in the nucleus. So there's plenty of GTP in the cytosol for this protein to take it up once it's dropped the GDP. But it can't bind both the GDP and the GTP at the same time because they bind to the same site on the protein. So if it drops it and then the GTP goes right into that active site. And now this is able to bind another charged tRNA. And the process repeats. What happens to that ribosome then? You now have two sites that are occupied. You have a P site and an A site that are occupied with a charged tRNA. That activates the peptidyl transferase activity of the large ribosomal subunit. And as we said before, the peptidyl transferase activity maps to the large ribosomal RNA in the large ribosomal subunit. So it's a 23S ribosomal RNA in bacteria. And it is the 28S ribosomal RNA in eukaryotes. So the large ribosomal RNA in the large subunits of ribosomes are ribozymes. That is, they are RNA molecules that have enzymatic activity. So the amino acid bound to the tRNA at the P site, it's transferred over to the amino acid attached to the tRNA that occupies the A site. You make a peptide bond between those two amino acids, and now this tRNA in the P site is empty. This tRNA now has two amino acids attached to it. What happens next is that a protein called EFG, which is also known as the translocase, comes and binds to the ribosome. Now, this is also a GTP binding protein. It binds to the ribosome, and by hydrolyzing GTP, it causes the ribosome to move one codon down the messenger RNA. So now the codon that was here at the P site now goes to the E site. The codon that was at the A site goes to the P site. The codon that was just outside the A site now goes to the A site. The ribosome is going to move down the tRNA in a 5' to 3' direction. And once you make a peptide bond and you have an empty tRNA at the P site, that tells the translocase to bind to the ribosome. Once it binds to the ribosome and hydrolyzes GTP, the ribosome moves. And now you have an empty tRNA at the E site. The empty tRNA has a very low affinity for the E site and it dissociates. That's why it's called the E site for exit. The tRNA exits. It will be recycled. It will be recharged by amino acyl tRNA synthetases, bound by a GTP bound EFTU, and be reused. So the tRNAs are constantly getting reused. And now you have an empty codon at the A site. And now EFTU bound to GTP will bring the next appropriate charged tRNA to the A site. You'll get two sites that are bound by charged tRNAs, and the peptidyltransferase activity will form a peptide bond between the nascent polypeptide that's on that one tRNA and transfer it to the other. And then the translocase will bind, and then it'll move to the next codon. And so on, and so on, and so on, and so on, and so on. That's elongation. Don't pretend this is simple, but it's how translocation works. It's how translation works.
You said the ribosome itself moves from side to
Yeah,
side?
the ribosome itself moves. It was thought for a long time that the RNA just got pulled through the ribosome. That turns out not to be the case. The ribosome actually gets up and moves. And a guy at my alma mater, UC Davis, John Hershey, has actually designed a way to videotape this. And he's got videotapes of ribosome moving down tRNAs using a very sophisticated labeling system where you can actually get live video of these things happening in real time. It's pretty amazing. And if you add antibodies to the translocase, the ribosomes, they just get stuck there, they can't move. And it's very, very cool. And you can make, you can use amino acids that are derivatives that are fluorescent. So you can see every time you put a new peptide bond, the amino acid fluoresces, and you can see the, it's really cool. I'll look for that video. Someone sent me a copy of it. I don't think it's on YouTube because it's John's proprietary property, but if I find it, I will show it to you. It's very, very cool. All right, so go back up here. We all clear about this? Go back down here. Stop it, you beast. Okay, we good with what's here?
I have a question.
Yeah, go ahead.
So the top one, that is showing the bigger picture of what they look like.
Yeah, that is a crystal structure of the ribosome.
And then the bottom one,
It's a cartoon.
okay, but it's the same thing.
They're showing the same system, yeah.
Yes.
Yeah. So this one shows the crystal structures of the protein, whereas that picture down there just shows a cartoon.
Okay, so then on so when the GTP hydrolyzes in the EFTU dissociation, that would be in the second step on the bottom one, right there.
Yeah, yeah, the charged tRNA bound to EFTU, bound to the GTP, is brought to the ribosome. You hydrolyze your GTP and EFTU leaves, and it's bound by EFTS. You exchange the GDP for GTP, and then it finds another dance partner, so to speak, and does it all over again.
Okay, so I'm just trying to figure out how the top and the bottom connect, just so I'm understanding it better.
This essentially is this cycle here.
Okay, so the top is just that little. circle.
Right.
Okay,
The top is more detailed than this part right here.
okay.
Yeah. That's how you get the charged tRNAs to the ribosome and have them associate with the right codon. Because you remember that anticodon loop we talked about in the tRNAs? That's the part that has to properly recognize the codon. So that 3-base sequence in the anticodon loop, that's what is interacting with the codon. If it doesn't interact properly, it's not the right tRNA, and it'll be rejected by the ribosome. If it is the right one, then it settles into position and the EFTU hydrolyzes the GTP and takes off. And loads back up with another GTP and gets another dance partner and starts to dance again. Yeah, again?
In the bottom part,
Mm-hmm.
in the like second to last one where the GTP is like connected, or the EFG is connected,
Yeah.
in the A site,
Mm-hmm.
what determines when that leaves and a new tRNA?
When the translocase leaves?
Yeah.
Once a ribosome is moved, its job is done. Also, it's hydrolyzed its GTP to GDP. Once it hydrolyzes its GTP to GDP, its affinity for the ribosome goes way down.
So then it just leaves.
It just leaves. It has to leave before the next tRNA can come because it's in the way.
Yeah.
So if it doesn't, ribosome doesn't move, then the translocase can't leave. And if the translocase doesn't leave, the next proper tRNA can't get to where it's going. Right? Now, this particular sequence is extremely similar in eukaryotes. It's just that the players are slightly different names. So EFTU and EFTS are just called EF1 alpha and beta. In fact, the EFTS is actually a 2-subunit thing called beta and gamma. So it's a 3-subunit ditty, but it does the same thing. Works the exact same way. So EF1 alpha is a GTP binding protein that brings charged tRNAs to the ribosome. And then the translocase in eukaryotes is just called EF2. EF2. So the mechanism is the same, the players just have slightly different names.
So the EFTU and EFTS is, what did you say?
EFTU and EFG and EF2 are analogous proteins. EFG is what you find in prokaryotes, EF2 is what you find in eukaryotes. Now, you might remember from last semester, we talked about something called diphtheria toxin. Diphtheria toxin is an enzyme that actually takes EF2 and it attaches a ATP group to one of its amino acids, and it completely inactivates it. So diphtheria toxin attacks this EF2, and it attacks it in such a way that EF2 is completely taken out of the picture. That's why diphtheria toxin is such an incredibly potent toxin, because it completely hamstrings translocation of the ribosome. That's one of the targets, essentially, for that particular toxin. But it doesn't do it to EFG, because EFG is a different enough protein that diphtheria toxin doesn't recognize it. All right, if you turn your handout over, here's what happens when you get to the end of no, here's what I want to do. I want to go like this. You're not going to let me get bigger. You should get bigger. Let's do this. View, let's zoom. Okay, can I do that? Oh, much better, much better. Now, if you get to a termination codon, termination codons do not have a tRNA whose anticodon loop matches their sequence. And you might remember that your termination codons include UAA, UAG, and UGA. Those are your three termination codons. Since you don't have a proper tRNA that binds to that site, instead, you have a protein called a release factor. So the release factor will also bind. Did I get the right picture? Doesn't look like I got the right picture, does it? Crumbs. Well, let me just explain it to you. So the release factor, the protein actually looks 3D like a tRNA. So it binds to that A site, but it doesn't have an amino acid attached to it. So the ribosome accepts it, but there's no amino acid there. Now, do you have two occupied sites as far as the ribosome is concerned? Yes, you do. So now when the peptidyl transferase tries to transfer that nascent polypeptide to the release factor, it clips the bond and then it just lets it go because there's no place to put it. And then the translocase binds. The translocase then tries to move the ribosome, and instead of moving the ribosome, it dissociates the two ribosomal subunits, and the ribosome lets go of the messenger RNA. Now, in eukaryotes, you have one release factor, and it's just called release factor. In bacteria, you have three release factors, RF1, RF2, and RF3, and they work together. And they specialize for different termination codons too. In eukaryotes, it's a one-size-fits-all sort of thing.
So the polypeptide connects to, like binds to the release factor?
Binds to the polypeptide when the peptidyl transferase clips one bond and makes another one. Uses the energy in that bond to make a new peptide bond. It will transfer that bond to another amino acid. The release factor doesn't have an amino acid attached to it. So it has a loop, there's nothing there. So the peptidyl transferase goes clip, nothing there. So it'd be kind of like a climber, essentially, who puts in 2 pitons to take his carabiners and put them through to put the rope through. He drives one piton in, he drives the other one in, clip, he gets the rope, and then he goes to clip the other one, and the piton is falling out. Oh, there goes my carabiner. Darn. Okay, I'll have to drive in another piton. That's what happens to the peptide. Peptide then is going to float into the cytosol. It's a new protein, and it's going to fold into its 3D structure, and then it's going to do it. So that's elongation, and that's termination. Neither termination nor elongation has quite as many steps as initiation. That essentially is how these work. Now, let's talk a little bit about there, that's a good size right there. So this peptidyl transferase activity that's in that large ribosome. What happens if you inhibit that activity? Right, you don't make peptide bonds, you don't make proteins. Turns out that several of the antibiotics we talked about last semester inhibit this very function. So for example, erythromycin and all the other macrolides, that's precisely how they inhibit protein synthesis, is they bind very, very close to the component of the peptidyl transferase activity and prevent it from doing its job. So macrolides in general Clindamycin, which is not a macrolide, also inhibits peptidyl transferase activity. And so does chloramphenicol, a drug that is really only used for emergencies in this country, is used in the third world, but is used in veterinary medicine. Why don't we use chloramphenicol? Does anyone remember that? Yeah, I know that was a while ago, wasn't it? Just wondering if anybody remembered. Causes aplastic anemia. It causes aplastic anemia in people at a rate of about 1 per 5,000 people, which is a high enough rate to ban it from being used in human medicine, except under emergency circumstances.
You tell us again what these do again?
These are antibiotics.
Yeah.
They inhibit the peptidyl transferase activity in bacterial ribosomes. So they inhibit elongation. Now, remember these? Who can give me an example of an aminoglycoside? They end with clindamycin. So, sorry? Clindamycin is not an aminoglycoside. Go ahead, Bree. Eomycin? Oh, neomycin. Yes, that's an aminoglycoside. Very good. Streptomycin, aminoglycoside. Gentamicin, aminoglycoside. Spectrinomycin, all those are aminoglycosides. They don't inhibit elongation. Instead, they cause bacteria to put the wrong amino acid in, and they also cause ribosomes to skip codons. So they cause the bacteria to make junk protein. And if you make junk protein as a cell, you're going to die, aren't you? Or at least you're going to stop growing. Remember that one? Tetracycline and its derivatives bind to the A site of the ribosome and keep tRNAs from being able to make contact with it and make protein. So they essentially gum up the ribosome and they prevent elongation from occurring. All right, so here are your stop codons. They're recognized by release factors. Release factors, at least their 3D structure, mimics tRNAs. And the bacteria have the three, RF1, 2, and 3. Eukaryotes only have one that recognizes all 3 stop codons, and then it has its partner in crime that helps it terminate. Translation is just called ERF3. And they bind to the A site and they fool the peptidyl transferase. The peptide is released, translocase binds, and the whole thing goes kablooey, essentially. Now consider this. Since bacteria don't have a nucleus, once the messenger RNA is made, ribosomes can jump on it right away. So very often, translation and transcription are occurring at the same time. That doesn't happen in eukaryotes because there's a nucleus that's in the way. Nuclear membrane that's in the way. So transcription and translation in prokaryotes is coupled. And that actually has rather profound implications for regulation. There are several types of genes whereby the gene is regulated by this coupling between transcription and translation. All right, so this just compares essentially the components of translation found in those 2 systems, prokaryotes, eukaryotes, and then here's a table essentially with some of the things that they inhibit when it comes to bacterial translation. Puromycin is a very powerful inhibitor of elongation. It inhibits translation both in prokaryotes and eukaryotes. Consequently, it's a very toxic drug. It's not used in human medicine, but it's extremely good for research. You want to shut down translation really quickly, puromycin is terrific for that. And so it gets used a lot in research. It's also a very inexpensive drug. That's one of the beauties of it. Are there any questions about transcription, translation, anything like that? Natalie?
So, for the translocase, does that, like, on a few slides ago, you said that it would like, once the translocase comes, then it stops, or like it goes off, is that what happened? Like, the translocase is kind of like, it's like,
During elongation or during termination?
termination.
The translocase helps with termination because it finds instead of the ribosome moving, the ribosome dissociates.
Okay.
And that is a function of the fact that the proper transfer to the tRNA that is at the A site doesn't occur. You have two empty tRNA, well, two seemingly empty tRNAs. You have a release factor that is mimicking a tRNA at the A site. That's why the whole thing falls apart. All right, this ends the material for your first exam. So what we will cover from now on will not be found on the first exam, and we will get into the textbook that I had you purchase. And this is largely about the medical use of genetics. And the goal of this was to just kind of get you up to speed and then show you how this applies in your practice at nurses, because you will encounter people with certain genetic diseases. There are certain conditions that are influenced by genetics. And so the goal is to help you get some background so that you can deal with people who have sickle cell disease, or some of you may work oncology. Genetics is a big part of oncology. Some of you will work mental health. Mental health is influenced a great deal by genetics, and so on and so forth. So we're now going to try to give you some Mendelian genetics so we can talk about inheritance patterns. Because very often in a medical history, you will be required to ask, or at least you should ask, the patient about their family history. And if they have a certain condition that it seems that they've inherited, you might need to ask them about their mother's side, their father's side, so on and so forth. And something you'll need to do is, after the patient has left, is to try and piece that together and make a pedigree. And if you need more information, you may need to ask the patient, find out for me about your deceased aunt who thought, she was General Grant, or something like that. Just, your grandmother who had this condition, or something like that. Pedigrees sometimes can be remarkably informative. Problem with pedigrees, you have to know how to read them. So I have some examples of pedigrees. So one of them is a pedigree for a genetic disease called juvenile hereditary glaucoma. And this one covers 7 generations of people in a family who have this condition. So glaucoma normally is a disease of the aged. This condition, however, appears quite early in someone's life. So if you look at this particular pedigree, you'll notice that there are a bunch of circles and squares and a bunch of lines. You're probably wondering, what does it all mean? Well, that's what we're going to try and explain today. So very often you'll be asked to make pedigrees with limited information. So sometimes you just might have things that you just have to put question marks sometimes. So we'll go ahead and we'll take a look at these pedigrees. From a pedigree, you can often infer an inheritance pattern. And we'll show you how we do that. So genetics very simply refers to inheritance, how things are inherited, why that inheritance works the way it works. When we talk about genomics, we're talking about the structure, makeup, gene content of the DNA that composes their genome. But genetics is how the genes encoded by the genome are inherited. So heredity results essentially from DNA molecules that are replicated and then assorted into gametes. Those gametes then unite when you became you, and those various chromosomes essentially were able to segregate independently of other chromosomes, and whatever combination of particular alleles that you came up with, that's essentially what you get. So genes will encode typically messenger RNAs. Those messenger RNAs get transported to the nucleus and then they're translated. And the proteins made by those genes and the RNA made by those genes, they will produce this thing called a phenotype. Phenotype is what you look like. What you look like. Genotype is what your genes look like. Phenotype is what you look like. Genotype is what your genes look like. Okay, as a little exercise, 10, 11 rats. We have an odd number of people. Turn to someone near you and take 10 seconds and describe each other's phenotype. And then take 10 seconds and the other person describe the other person's genotype. Go. Out in the North 40 there, Emily. Maddie's right there. So. Okay. All right. I think that's enough of that. Did you exchange enough information? That's the way you look. That's your phenotype. That's your phenotype. Now, if I asked you to do the same thing and say, describe each other's genotype, what would you do? You look at me askance and say, are you high? Because you can't see their genes, can you? You can infer their genes based on their phenotype. But to know someone's genotype, very often, you have to look at their heredity patterns. Their heredity patterns, or you can have their genome sequenced, which is a lot more expensive. One or the other. So genotype, that's the way your genes look. Phenotype, that's the way you look on the outside. So when you look in the mirror and you look at yourself, that's your phenotype. Will your phenotype change as you age? Yep, unfortunately, it happens to all of us. This used to be kind of brunette. Now it's all gray. So my wife had hair very much like Caitlin's, and then now she has hair a lot like Sydney's. She says there's no gray in it, but I'll bet if I look hard enough, I can find some. Anyway, don't tell her I said that, by the way. It happens. You change, your phenotype changes as you age. And aging is also something that is linked to genotype as well. So genes are assembled on linear molecules of DNA called chromosomes. When you think of a chromosome when it comes to transmission genetics, a chromosome is a unit by which genes are transmitted between generations. It's a unit of genes that are transmitted from one generation to another. So humans have 23 chromosome pairs, 26 Well, the slide comes from the book and they say chromosomes. That's actually not quite right. It's chromatids, but we won't begrudge them that now, will we? One of the pairs of these chromosomes is the so-called sex chromosomes. Sex chromosomes. The X chromosome, which is relatively large, the Y chromosome, which is pretty small. Females have 2 X chromosomes. Males have an X and a Y chromosome. The remaining 22 chromosomes are called autosomes. Autosomes. That's your nuclear genome. 22 autosomes, 1 pair of sex chromosomes. And then you have an additional genome in your mitochondria. Mitochondrial genomes are small. They only encode a few proteins. Having said that, mutations in mitochondrial genomes have profoundly deleterious effects. So let's talk about genes on chromosomes. So let's say we have, a chromosome. Let's say on this chromosome, we have a gene. This area on this chromosome that encodes that gene is referred to as a locus. A locus. And the type of that gene that you have there is referred to as an allele. An allele. So if I had, say, 7 different types of paper, one of those types of paper, say, was red, and had yet another type of paper, and it was green. Third type of paper, that was blue. Fourth type of paper, that was orange. Another type of paper, that was violet. Another type of paper, that was black. And then the 7th type of paper, that was white. These are all paper, right? But they're different types of paper. They're different alleles of paper. That's one way I think to think of alleles. They're types of genes. You all have genes that determine skin pigment. Some of your alleles produce this much pigment, and you're on the pale side of the fence. Some of you have alleles that make a touch darker. Your hue is slightly darker. Others have alleles that make a bit more, and some have alleles that make even more. That handful of genes that determine skin color come in different types. Some make a lot of pigment, some make not very much pigment. Those are different alleles of those genes. So if you have on your chromosome pair at the same genetic locus, the genes found at that locus, they have the same alleles. Let's just call them something. Let's call them big A on one chromosome, right there, That's big A. Another chromosome. This one also has big A. The two alleles are the same at those genetic loci. This arrangement is called homozygous.
What does it mean in the word that
Homologous. Homologous means these are the same copy of that chromosome. So if this is chromosome 1, that's the chromosome 1 you got from your dad. This is chromosome 1, that's the chromosome 1 you got from your mother. They're both chromosome 1, but they're not exactly the same. But they're similar enough to be identified as chromosome 1. Those are homologous chromosomes.
So when you say copy, you're not like saying it's identical. You're saying
No.
it was made from the same.
No. That chromosome that was in your mother's egg, that's a maternal copy of chromosome 1. It has the alleles that your mother had. The other one was in the sperm, the pronucleus of the sperm that came from your dad. And when they were intimate, they made you. It was due to the fusion of those two cells that produce this unique copies of a maternal copy of chromosome 1, paternal copy of chromosome 1, maternal copy of chromosome 2, paternal copy of chromosome 2, maternal copy of chromosome 3, maternal copy of chromosome 3, all the way up to the chromosome 22 and then the sex chromosomes. So these chromosome 1s are homologous chromosomes. The chromosome 2s, maternal, paternal, those are homologous chromosomes, so on and so forth.
What is it called when two chromosomes are together? Like, do they come together, don't they? Or no?
Yeah,
So what is that called? Just a chromosome?
during meiosis or just during interphase.
Never mind.
When the two chromosomes are paired together, they're typically just referred to as a chromosome. When they're separate like this, they're usually referred to as a chromatid, but the two chromosomes in the pair are called homologous chromosomes. This is often called a sister chromatid. So they're sisters. Aren't they brothers? I have no idea. They're called sisters. Question, Katie? Is that a question?
We were just talking.
You're just talking?
Yeah, we were just sister
No.
chromatids chromosome.
Yeah,
That's what we said.
tid, so, yeah. And that's what's confusing is that the two sister chromatids are sometimes referred to as homologous chromosomes. So that's a little confusing.
So that's why you said 46 chromatids.
Right, but 23 chromosomes,
That makes sense.
right. 46 sister chromatids. Now let's say over here, we didn't have that allele. Let's say we had this allele. So we have two different alleles at those genetic loci. That situation is called heterozygous. So let's use a very simple example using this particular set of concepts. Say we have two people and both of them are heterozygous for the allele for cystic fibrosis. So this person, there's one chromosome, there's a wild type copy of the cystic fibrosis gene, the CFTR gene. The other copy of the chromosome has a mutation in the CFTR gene. If there's two copies of that, they will have cystic fibrosis. So this person as children with someone else who's also heterozygous for this particular cystic fibrosis allele. Now, they may not even know this. What are the chances that their child will have cystic fibrosis? It's a very easy way to figure this out. It's called using a Punnett square. This was designed by an American geneticist named E.B. Punnett. Punnett squares are taught in high school biology textbooks. You may have seen a Punnett square before, but it is by far the easiest way to do these types of problems. What you do is you separate the gametes. So you take, let's say this is dad, let's say this is mom. So dad's gametes go here. So that's dad. Mom's gametes go here. And then you just do the cross. So this person here, that person will not have cystic fibrosis, because cystic fibrosis requires 2 of those alleles in order for the person to have the disease. This person here and this person here will be heterozygous. They will not have the disease. This person here, that person will have the disease. So 1 of 4 possibilities, so there's a 25% probability that their child will have cystic fibrosis. And that falls out just by doing it that way. So Punnett squares are your friend. Don't say, oh, I'm not in high school anymore, I can't do Punnett squares. I do Punnett squares all the time.
So then for the heterozygous, that's like, they could have cystic fibrosis or they won't?
No, they're carriers of it. They're carriers of it. They're heterozygous carriers of it. All right, we'll pick this up on Monday, 28th, 28th, I think it's 28th. Yeah, it's on a Monday. Figured if you had the weekend to study, that would probably work well.
So when do you want us to get questions to you by then?
Definitely by the weekend. Because that's probably when I'll write the exam.
So we
Okay.
can make questions.
Yeah,
for the
go ahead,
exam?
Seth.
So we can make questions for the exam?
Oh, yeah, yeah,
Okay.
definitely.
Okay. All right. Thank you.
According to Bree Vilmont, they're doing a Google Doc. Do you want Bree to send you the link?
Yes, please.
Oh, wait, McKenna made it. McKenna will send you the link.
Okay.
Did you hear that?
No.
Say it again.
Are you in the group chat for Snapchat?
No, I'm only on Facebook.
Okay, I'll send it to you on there then.
Okay. All right. Thank you.
All right.
All right.
Have
Thank you.
a good weekend. How you feeling?
I'm good. I went to the ER on Tuesday
Mhm.
to get everything checked up and everything was fine.
Good, good,
I
good,
just
good.
didn't
Okay.
feel well.
Look forward to seeing you on Monday then.
Yep.
Have a
All
great
right.
weekend.
You too.
Bye.